As a potential sample contributor to the 1KSA project, you may be interested in finding out more about the 1KSA workflow. The first step is to access the 1KSA website where you are now to find out more about the project, specifically about the DNA requirements and the terms of use of the project. You can also fill in the species nomination form on the 1KSA website. This form will be submitted to the steering committee for 1KSA, which constitutes a representative of the 1KSA labs. This group meets once a month in order to consider nominations, and if you are successful, you will receive an email from 1KSA to this effect. You will also be invited to submit an online sample submission form. These entries are collected on the 1KSA REDCap project database. Once you have submitted your sample, the next step is DNA submission. DNA can be submitted to one of several 1KSA labs, and these labs will then carry out library prep and sequencing on the PromethION. The raw sequencing data is then transferred and uploaded to the NICA system, and a draft assembly pipeline is then carried out on the CHPC. Both the draft genome assembly and the raw sequencing data is curated and stored on DARISA. Through DARISA, the data is then made available to the sample contributor, and the sample contributor can use this data automatically for 3 years. However, we do request that permission is obtained from the Data Access Committee before publication, and I'll talk a little bit more about that in a few clicks. During the sequencing and the draft genome assembly steps, a number of different metrics are recorded, and these are also uploaded onto the REDCap project database. Following completion of the draft genome assembly, a project report is generated from the data in REDCap, which is then sent to the sample contributor. This is a list of the methods used in the sequencing and in the draft genome assembly, and it is a record for future publication purposes. Separately, a 1KSA species card is made for the species of interest, and this is uploaded onto the 1KSA website. So the 1KSA website is not only the place where you can nominate a species for sequencing, it's also the place where you can get more information about the project. And very importantly, it's also going to be the vehicle for highlighting visibility around the different species that have been sequenced under 1KSA through the different species cards. It might come to pass that a second scientist is interested in the same genome, and they will be able to then request access to the data through the Data Access Committee. This committee will be set up by 1KSA, and the idea is that the Data Access Committee will consult with the sample contributor for the first 2 years after sequencing, and they will also have overview of the curated and stored genomes and draft genome assemblies on DARISA. If the Data Access Committee decides to grant access to the scientist, it will be done through a data transfer agreement, which will detail the very specific conditions under which the scientist can use the genome sequencing data. Over time, we expect that both the sample contributor and any additional scientists may generate commercial intellectual property from the genome sequence. We don't know what the timeline will be on this, and so the 1KSA project will request that the scientist and the sample contributor respectively will keep track of this commercial intellectual property, and that over time a portion of the royalties accruing from this intellectual property will then be contributed to the 1KSA Educational Trust. This trust will be set up by 1KSA in order to promote biodiversity genomics education, and in particular it will be used for scholarships, which will be awarded to learners coming from rural areas in which the original species sequenced under 1KSA would have been collected. If you have any further questions, please reach out via our contact details. Thank you.